How do B lymphocytes become plasma cells and memory cells in acquired immunity? So in order to understand what is involved with acquired immunity, it's important to understand the relationship between innate immunity and acquired immunity. So the immune system is a mechanism, a system within the body that protects the body against pathogens and materials that are going to be harmful to that. And so within immunity, we've got two forms. We've got innate immunity and acquired immunity. Innate immunity is going to be immunity against a generic category of things. So innate immunity would be against bacteria. And so things like macrophages are going to recognize the bacteria as being potentially pathogenic and will attack it in a kind of generic manner. In contrast, acquired immunity is going to be an immunity against a specific pathogen. And so it may be against a specific virus or a specific type of bacteria. And that is generally going to be associated with antibodies, which are molecules that are going to be effective at recognizing the specific shape associated with that pathogenic material. Okay, so the antibodies are going to be examples of immunoglobulins, and so they're going to be globular proteins associated with the immune system. And we're going to see that it's going to form a Y-shaped molecule comprised of two heavy chains, kind of forming the full structure of the Y that we're seeing in here, as well as two light chains, giving us this kind of composite multi-polypeptide chain protein. Now, in this Y structure, there are going to be two regions that are going to be incredibly important. The first region is going to be the tips of the Y. The tips of the Y are going to be highly variable based on the plasma cell, the B lymphocyte that ultimately is going to be producing the antibody. And so we're going to have a lot of variability within the antigen binding site at the tip of the Y. And you want that because you want to be able to recognize specific types of antigens. You want to be able to identify the specific pathogen involved. Okay, so you've got the highly variable antigen binding site at the tips of the Ys, and then kind of the base of the Y and the kind of the stalk of the Y, the shaft of the Y, kind of in this region right through here. This is going to be the constant domain, and so there are going to be a few different types of constant regions. Again, a limited amount of variability, but the variability associated with the constant domains is going to be determining what are the characteristics of this antibody. So is it an antibody that is going to be able to pass across membranes? Is it an antibody that forms multiple structures? Basically, is it going to be involved with kind of directing certain types of cells to respond to eliminate that pathogen from the body? So in a generic sense, what is going to happen is that our B lymphocyte, and so this is going to be a naive B lymphocyte, is going to have some receptors along its surface that are going to be very, very similar to the antibodies that they're going to be able to produce. And basically what happens is that once those receptors on the surface of the B cell bind to the antigen, they're going to engulf, basically the B cell is going to engulf a little bit of that antigen and then express it on their surface in relationship to MH class II molecules. And so basically this is an antibody that is able to recognize a specific antigen type, so a specific pathogen, based on the shape and based on its interaction with this B cell receptor. Now what's going to happen is that our helper T cells are going to come along and they're going to recognize that this B cell now has the ability to recognize a pathogen that's found in the body, so be able to recognize an antigen that's present within the body. And so this T helper cell is going to recognize that basically the antigen shape in relationship to the MHC class II molecules, and they're going to activate this B cell. And so they're going to stimulate this B cell to start to divide. As a outcome of dividing, we're going to have more copies of the cells. Some of those cells are going to become short-lived plasma cells, and other cells are going to stay relatively undifferentiated. They're still going to be able to produce that antibody, but they're going to serve as memory cells. So you have more cells with a subsequent exposure to that pathogen that are going to be able to respond to it. So what we can see then is that our B cells are going to be very important for our primary and secondary immune response. And so this is why immunizations are important, why vaccines are important. And so basically on this diagram, along our x-axis here is going to be time, along our y-axis is going to be antibody concentration. So what happens during your initial immune response, so a primary immune response, is that you're going to have naive B cells that are capable of responding to the antigen but have not seen it before. They're able to respond to the exposure to that antigen. They're going to go through a process of cell division. Some of those cells are going to become short-lived plasma cells, and they're going to be able to produce antibodies. Other cells are going to be memory cells that are going to be a reserve population in the body that can respond to future exposures to that antigen. So your primary immune response is basically going to be slow and limited because you have few cells that are capable of responding to that. And so you see this kind of initial kind of response coming up here, kind of a mild production of antibodies, but over about 2 to 3 weeks, that's basically going to go away because of these short-lived plasma cells. With a subsequent exposure, and so this may be a booster shot if you're looking at a vaccination, or it could be exposure to the pathogen. You've now got these memory B cells that are capable of responding to the antigen, and you've got more of these memory B cells than you had of the initial naive B cells. And so the secondary immune response and subsequent immune responses are going to build upon the fact that we've got these memory cells present. And so we're going to see a much more rapid response, a faster kind of uptick in our production of the antibodies because we've got more of these memory cells that are going to become activated, kind of similar to what we had previously here. They're going to bind to that antigen. Our T helper cells are going to say, okay, time to do it all over again. Well, not over again because they're memory cells. They didn't undergo that in the first place. But basically, they're going to activate these memory cells to divide, some of which are going to become plasma cells. More cells are involved, so they're going to be able to produce much greater quantities of antibodies much more rapidly than our initial primary immune response. And so that is often going to result in kind of a lasting level of antibodies within the body for a prolonged period of time. Now, in addition to these plasma cells, we are also going to have more memory cells that are produced so that with each subsequent exposure to that pathogen, you're able to mount a very vigorous, very rapid, very strong immune system response to that using these antibodies being produced by the plasma cells that are generated by activation of these memory cells.